Desmosomes are adhesive or anchoring variety of cell junction. These are also known as macular adherents, and they are seen in the tissues which experience maximum stress. Desmosomes are located in cardiac muscle tissue, urinary bladder tissue, gastrointestinal mucosa, and stratum spinosum of epidermis. Structurally, these desmosomes are highly organized and they are electron-dense structures. In the desmosomes, the inner aspect of the plasma membrane of adjacent cells have disc-shaped proteins bearing protein fibers known as keratin or intermediate fibers, which extend into the cytoplasm, and they are connected with the cytoskeleton of the cell. Desmosomes are made up of intermediate filament complexes, then network of cadherin protein and linker protein and keratin. Desmosome intermediate filament complex has got three parts, as we can see in the figure. The extracellular core region is formed by desmoglia. So it is a cell adhesion protein present in the extracellular core. We have mainly two proteins, desmoglein and desmocholine. Then if we see the plasma membrane, there is a plaque formation and there is outer dense plaque and inner dense plaque. Inner dense plaque is facing towards the cytoplasm and the outer dense plaque is facing towards the extracellular space. So the outer dense plaque mainly contains intracellular ends of desmocholine and desmoglein, and it also has a protein known as desmoplakin, as it is labeled in the diagram. You can see the inner dense plaque contains the terminal portion of desmoplakin and their attachment to keratin intermediate filaments. Functions of desmosomes are: the desmosomes are binding proteins. They hold the tissues together. Then they provide anchorage sites for intermediate filament and thus they maintain tissue integrity. They also affect the cadherin protein based signaling pathway. They connect the intermediate filaments to the cytoskeleton of adjacent cells. They have got clinical importance also. The arrhythmogenic cardiomyopathy is caused due to mutation in desmoglein 2 gene. Also, blistering autoimmune diseases like pemphigus vulgaris is caused by circulating autoantibodies immunoglobulin G that target to the desmoglein 3 protein. In this diagram, we can differentiate the desmosomes from hemidesmosomes. So, the desmosomes form cell-to-cell adhesion. We can see this is one cell, this is other cell, the plasma membrane and intercellular space. So, the cell-to-cell adhesion is present in desmosomes, whereas hemidesmosomes form the adhesion between cell and plasma membrane. So we can see this is the basal part of the cell and this is the basement membrane. So the adhesion between the cell and the basement membrane, this is the hemidesmosome. The proteins involved in formation of desmosomes are mainly desmoglein and desmocholine, whereas in the hemidesmosomes, we can see the proteins are plectin. This plaque is formed by plectin, and these anchoring are the integrin proteins. Then, if we see the location, desmosomes are commonly located in cardiac muscle tissue and gastrointestinal epithelium where maximum stress is there, and hemidesmosomes are present in the keratinocytes. Then the function of desmosome is cell-to-cell integrity and rigidity, whereas hemidesmosomes provide adhesion of the cell to the basement membrane. The clinical significance of desmosomes is mutation in the desmoglein protein causes arrhythmogenic cardiomyopathy and blistering diseases like pemphigus vulgaris, whereas the applied importance of hemidesmosome lies in muscular dystrophy and epidermolysis bullosa. It occurs due to mutation in the genes mainly integrin and plectin.